Hello everyone, welcome to my YouTube channel. Today we'll be discussing a topic called vectors. Vector can be defined as a biological tool in recombinant DNA technology. Vector is used for the delivery of desired foreign DNA or gene of interest into the host cell. Vectors are the DNA molecules which can carry a foreign DNA fragment to be cloned. The most important vectors are plasmids, bacteriophages, cosmids, and phasmids. Coming to the classification of vectors, based on the nature and sources, the vectors are categorized into following classes: plasmids, bacteriophages, cosmids, phasmids, shuttle vectors. For time being, we'll be ignoring shuttle vectors and discuss the rest of the four vectors. Coming to the first vector, that is plasmid vectors. Plasmids are extrachromosomal, double-stranded, circular self-replicating DNA molecules. Almost all the bacteria have plasmids containing a low copy number 1 to 4% or a high copy number 10 to 100%. The size of plasmids varies from 1 to 500 kb, that is kilobases. Usually, plasmids contribute to about 0.5 to 5% of the total DNA of bacteria. Examples of plasmid vectors are PBR322 and PUC18 or 19. PBR322 vector is one of the most popular vector, and the representation of PBR322 vector is shown. It contains different restriction sites such as ECO-R1, HIND3, and many other PAM, H1, and even it consists of antibiotic-resistant genes, ampicillin and tetracycline. Coming to bacteriophages. Bacteriophages are the viruses that replicate within the bacteria. In case of certain phases, their DNA gets incorporated into the bacterial chromosome and remains there permanently. Phage vectors can accept short fragments of foreign DNA into their genomes. The advantage with phages is that they can take up larger DNA segments than plasmids. On the basis of structure, bacteria phages are of two types: head and tail phages and filamentous phase. The best example for head and tail phases is lambda phase, and for filamentous phase, it is M13 phase. The size of lambda phase vectors is about 48,502 base pairs. It contains origin of replication, genes for head and tail protein, and enzymes for DNA replication. It should be larger than 38 kb and smaller than 52 kb to pack into phage particles. It has more than one recognition sequence for almost all the restriction enzymes, and that is the specialty of bacteriophages. Coming to cosmid vector, it is a type of hybrid plasmid. Simply, it is defined as a combination of cos site plus plasmid. It contains lambda phase cos sequence. Genomic size of cosmids is about 30 to 52 kb. They have suitable origin of replication that they can replicate as plasmid within the host cells. Examples, as you can see, SP40 ORI. It also contains selectable markers such as ampicillin resistant gene. That means it has resistance towards the antibiotic, that is ampicillin. Collins and Horn in the year 1978 was first to describe cosmids. And the final and foremost vector is phasmid vector. So, what is phasmid vector? A phasmid vector can be defined as plasmid vector which contains an origin of replication from a phase in addition to that of the plasmid is termed as phasmids. And here P blue script SK, which is positive or negative, is a type of phase-mate vector of 2958 base pairs derived from the PUC 90. Thank you.